Yes, so thank you for that introduction. And I'm very grateful to have been invited here today to speak to you by the TBA. In particular, because it's not often, as I spend most of my time holed up at the University of Cambridge, that I get to actually interact and speak with people to whom my research is directly relevant. And I'm hoping at the end of the talk today, I will have captured your interest in my subject of research, and I would be really grateful as well for any feedback on any of the ideas that I put forward towards the end of the talk. So today I'm going to be talking about my research project, which is co-funded by the Horse Race Betting Levy Board, the TBA, and the European Breeders Fund. And what I'm trying to investigate is a completely novel approach to worm control, whereby we can exploit interactions between parasites and gut bacteria. And I apologize in advance because whoever decided to name bacteria wasn't very sympathetic to either the presenter or the listener, because a lot of the names are quite unpalatable and probably mean nothing to people, but I will try and keep it simple and make some general points. And if I do lose anybody at any point, then please do put your hand up or come and see me afterwards, because I haven't done my job properly today if I haven't got the main message of my research across. So first of all, just going to go over some brief background. So we all know that the cost of worms on thoroughbred stud farms, and in my mind this can be broadly divided into two main issues, and one of them is the underlying effect of worms on growth, particularly in young animals. And it's inevitable that an animal with a high level of parasites, it's going to have a negative impact on their nutrition and on their growth, and therefore their condition, and when it comes to sales. And also, occasionally, when parasites aren't well controlled for whatever reason, we can see some quite nasty, severe adverse effects of having high levels of worms, particularly in young animals. So particularly, these can be attributed to a high level of small strongyles, which can result in a condition known as larval cyatostominosis, which is basically protein-losing diarrhea, which can be fatal. And also ascarids, high levels of ascarids in young animals, particularly foals, can cause complete blockage of the intestine, which results in them needing surgery for colic, which is obviously life-threatening. So fortunately, these events are fairly rare, especially when parasite control regimes are in place on stud farms, but still we need to be aware of the possibilities of losing control of these infections. So unfortunately, since their introduction in the 1940s, parasites have developed resistance to worming drugs systematically in the space of about 5 or 10 years of their introduction, although ivermectin or Ecfelan, with the name of the product on the market, has actually lasted a lot longer than that in horses, but we're currently now seeing resistance developing to that as well. So the reason I wanted to point this out was because we can't rely on the fact that we're always going to have drugs available which work. I mean, a lot of these drugs have very different mechanisms, and the fact that worms have developed resistance to all of them should highlight that worms are really brilliant at developing drug resistance, and therefore, in the future, even if we get another product coming out in the next few years, we're probably going to get resistance to that in the future. So we can't totally rely on drugs to control parasites. And here I just want to highlight what the current resistance status is to the various worms available in the various parasites which affect horses. So sorry, this isn't a very aesthetically pleasing table, but the green ticks just show that these drugs are still reported to work in some situations or in most situations, and the red cross is evidence of there having been resistance reported to the drugs. So as I've made apparent in the first slide, and you know that ascarids and small strongyles are probably the most worrying and pathogenic parasites that we have on stud farms in young animals, because the large strongyles don't appear to have any resistance to drugs. They're very, very sensitive to wormers, and also tapeworms, the drugs appear to be effective, continue to be effective to them. So with ascarids, in the UK, benzimidazole or Panacur continues to be a very effective drug to treat these infections, although worryingly there have now been reports of resistance in other countries such as Australia. And so we can't, the fact that we use them so frequently means that we can't continue to necessarily rely solely on this group of drugs to control these worms, but it is our main mechanism at the moment to control them. When it comes to the ivermectin and moxidectin wormers, unfortunately, there's been quite a wide degree of reported resistance in ascarids to these, so people tend to not use these as a mainstay of control for ascarids. And parental, there is resistance, but it also can still work. For the small strongyles, you can see here that there's pretty much no point using benzimidazole to control these infections because resistance is so widespread. However, ivermectin has continued to be effective for many years, although we're now starting to see some reports of complete resistance, especially in countries like Brazil. And also, what we're seeing in this country is that although the drugs are still working, or it appears they're still working, we're getting eggs coming back much faster. So clearly, the drug is starting to fail, but we're just not seeing it clinically yet. So my main point I want you to take away from this slide is that what we mustn't do is just rest on our laurels and assume that benzimidazoles work against ascarids and ivermectin works against small strongyles, and we should just continue to use them in that way. What we need to be doing is checking on farms for resistance before using a drug to specific parasites, and so that you're treating for the specific parasite you want to kill with the right drug at the right time, so that we don't continue to make this problem worse. So other methods which have been widely advocated in recent years to try and reduce our reliance on drugs and reduce selection pressure for resistance are using targeted treatment. So this basically describes just testing fecal egg counts in animals. It's very simple, and then you treat animals with a high fecal egg count, because what you find is that most animals in a herd, particularly in older herds, will have a low fecal egg count and don't need treating, and so there's no point using drugs against them because all you're doing is making the resistance worse while not actually improving the health of the animals. And also what this does is, because the majority of the animals which have the high fecal egg count are going to be putting eggs out onto the pasture, they're going to be the main source of infection for the other animals in the herd. So by treating this small group of animals which have high levels of infection, you're also hoping to reduce the overall infection levels in the herd. And also there are other methods such as management. Removal of droppings is very effective, but unfortunately can be quite practically difficult when you've got a large amount of animals on a large area of land. So it isn't always possible to implement this. But what I want to really focus on today, and what my research focuses on, is looking at alternative approaches that we can use not to eliminate the need for drugs to control worms, because we evidently, especially in young animals that are kept in close quarters with each other, we're inevitably going to have to use drugs to stop them from getting severe adverse effects of having very high levels of parasites. But there may be other approaches we can use in the herd as a whole that we can advocate in order to reduce the overall need for drugs to sort of help all of the animals to prevent the negative effects of parasite infection. And I want to point out what I think are quite startling numbers, that horses and worms have evidently co-evolved over hundreds of thousands of years together, and the horses by and large are absolutely fine and don't succumb to parasite infections regularly in natural environments. And we've only had wormers for 70 years. So clearly, you know, there are other factors at play which determine whether an animal can tolerate a parasite infection. It's not just getting rid of it with a drug. There are other important factors. So this horse here is a conic horse, and they live in Wicken Fen, and I was fortunate enough to be able to do some research on these ponies a few years ago. And this is a 2-year-old horse which has a fecal egg count of about 5,000, which you would balk at if you saw that in one of your yearlings. But it's absolutely fine. In fact, it's overweight. And the reason for this is because this horse probably has adequate nutrition, its immunity is developed, so it can fight off having these adverse effects of parasite infections. And so what I want to highlight here is that the impact on the animal, and therefore the need to give a wormer, is sort of a balance between the number of parasites that it's getting infected with at that point in time and its nutrition and its immunity to those infections. And the focus of my research is really on the nutrition and immunity side. And so the questions that I sort of wanted to try and answer with my research was, firstly, what impact do worms have, what exact impact do worms have on nutrition and metabolism? Because we all know or think we know and assume that parasite infections have a negative effect on growth and weight gain. But why is that? And is there anything that we can do to counteract this? And secondly, what impact does nutrition have on immunity? And I think that as more and more research is done in other species, especially in humans, it's becoming really evident that what we eat and what we put in our body has a huge impact on our immune systems and our overall health. It's not just a simple system that we're dealing with here. So the other question is, can we therefore use nutritional or dietary interventions to improve immunity to infection? And in order to examine these questions, I chose to look at another group of organisms which are closely linked to both worms and horses, and these are the gut bacteria. So as a horse relies on intakes a plant-based diet, it relies on its gut bacteria in order to produce most of its nutrients in order for it to grow and survive. And we know that the gut bacteria also has a huge impact on your immune system, especially during your development as a young animal. And so it's entirely plausible to suggest that the gut bacteria and their interactions with worms may be involved, or the interactions of worms with gut bacteria may be having a negative impact on the host animal. And that's why I'm chosen to sort of focus on whether or not worms and gut bacteria interact with each other. So the overall aim of the study, although there are several parts, I'm just going to be presenting one part today, is to compare the gut bacteria between animals, thoroughbred yearlings specifically, with high and low parasite burdens. And I've chosen to do this in the small strongars because they're widely prevalent and it's quite easy to select reasonably sized groups of animals, some of which have a high fecal egg count and some of which have a low fecal egg count, because that's what I needed to do to compare the gut bacteria in these animals. So this is how I performed the study. So basically I took a group of yearlings and screened them using just sensitive fecal egg count technique and divided them into two groups based on having either a high fecal egg count, which I classified as over 100 because actually overall the fecal egg counts weren't very high at the time that I did the study, and also a low fecal egg count, which was less than 10 and usually 0. And then I took fecal samples from the animals at the beginning of the study and then treated them and repeated the fecal samples. So in order to look at the gut bacteria, because we know now that a lot of the bacteria which reside within your gut can't actually be grown in a laboratory because it's just been possible to grow them, what we did was extract the DNA from the fecal samples and just sequence it to see which bacteria were there. So this generated a large amount of data, but it basically essentially was telling us in each fecal sample that I got from each animal which bacteria were present. And these are some of the results. So these are just the fecal egg counts showing that there was a difference between the high fecal egg count and the low fecal egg count group. And this graph here is a graph to show, so each dot is a horse, and it's a graph to show the overall composition of the gut bacteria of each animal and how it relates to that of the other animals in the study. And what you can see here is that the animals with a high fecal egg count definitely separated into a distinct group from the animals with a low fecal egg count. So this is suggestive that parasite infection or high levels of a parasite infection in these animals was leading to an alteration to their gut bacteria. And now I just want to digress slightly to talk about one of the concepts which we use to describe gut bacteria, because it's important when I come back to my results later on in order to, so you can fully understand the meaning of the results. So what we sort of get quite interested in with gut bacteria, as well as which bacteria are there, is this concept of the diversity of your gut bacteria. So this can be divided broadly into two measurements, and one of them is richness, which is basically how many bacteria you have in your guts. So if you've got lots, you've got a rich gut bacteria. And also there's the concept of evenness, which basically is the how evenly distributed the levels of these different bacteria are. So have you got loads and loads of one and hardly any of the others, or have you got, are they all sort of relatively evenly abundant? So this diagram just demonstrates this with fruit. So you can see here, imagine this is a fecal sample, that you've only got 3 types of fruit and you've got loads of apples and only 1 pear and 1 orange. So this has got a low diversity because it's a low richness and a low evenness. Whereas this sample here on the right has got lots of different types of fruit, and there's roughly the same number of each of them, which means that this sample has a high diversity. And why this is important is because a lot of studies that are now coming out in a lot of different species show that often a high diversity is associated with health. And when animals get sick, or animals which even before they get sick, as a risk factor for becoming sick, they have a low bacterial diversity in their guts. For example, children who develop asthma have been shown to have a lower bacterial diversity when they're born. So in the first days of their life, they've got a lower bacterial diversity, and that is a risk factor for them then going on to develop asthma. So it's actually really quite important, although there's sort of mechanisms behind it we don't really understand at present. But what I saw in the yearlings was that, and the animals which had a higher fecal egg count had a lower richness. So this is suggestive that the parasite infection was having a negative effect on their gut bacteria and causing the diversity to drop. And what we usually use as a term to define a negative effect on gut bacteria is it was causing a dysbiosis or a disruption to the gut bacteria. And what was interesting was that after treatment, the richness increased. So you could see that removal of the parasite then reversed the effect, and you saw an increase in richness, which kind of gives me confidence that this was a change which was genuinely associated with parasite infection. Now, I then went on to look at which bacteria were responsible for these changes, so which specific groups of bacteria. So this is when we get into a lot of names, which doesn't really matter what they are, but I'm just going to make some overall points. And this is a network analysis. So each dot is a type of bacteria, and the ones that are red are linked to animals with high fecal egg counts. So they're higher in the animals with high fecal egg counts. And the ones which are blue are the ones which are higher in the animals with low fecal egg counts. And what we saw was that there was a distinct group of bacteria which were quite closely related to each other, which went up in infection. So, for example, the Mogibacteriaceae and the Clostridiaceae. And then there were another group which seemed to go down in infection. And what was even more interesting was that when these animals were treated, you saw a reversal of these effects. So the bacteria which were, so this is the same analysis, but instead of this time, the red is before treatment and the blue is after treatment. So before treatment, you see the same bacteria which were linked to infection are higher. And then after treatment, the bacteria which were put down by infection go up. So it's quite nice that you can see this reversal of effects by treatment, which gives me confidence that these bacteria are genuinely associated with infection. And just, I want to talk a bit more about what these bacteria are and how they relate to each other. So what was even more interesting was that the group of bacteria which went up in infection all belonged to the same big group of bacteria, the firmicutes, and most of them belonged to Clostridia, the class Clostridia. Whereas those that went down with infection belonged to the class Bacteroidetes in phylum Bacteroidetes. So the fact that these bacteria that were going up and down were biologically related to each other is also sort of interesting because it makes you believe that there's something genuinely going on here. And this is just one example of a family which was strongly associated with infection called Paraprevotalaceae. And this is the low fecal egg count animals in red and the high in blue. So you can see that the levels are lower in the animals with high fecal egg counts. When you treat them, they go up, whereas if you treat the animals with low fecal egg counts, there's no change in the levels of this bacteria. So this, again, is more evidence that these are genuine links with infection. The reason I'm emphasizing is because a lot of studies looking at gut bacteria show things which may or may not be true associations, because if you just take one snapshot, you might see one bacteria going up just by chance. But the fact that these are reversed with treatment makes me sort of more confident in these results. So having sort of shown that these specific groups of bacteria are going up and down with infection, I then wanted to look at what the potential implications of this were. So in order to do this, you can use the data from the sequencing to predict what the function is of the gut bacteria, and therefore what metabolic pathways are likely to be up and down regulated as a result of the presence or absence of specific types of bacteria. So what this shows is a before and after treatment, the different pathways which were up. So before treatment, you can see there are very few pathways which are increased. And these are all related to short-chain fatty acid metabolism, which are actually, which the Clostridia, which were up with infection, are actually quite known to be involved with. So this made sense. 2 days after treatment, you see an increase in a lot of pathways which are involved in carbohydrate metabolism, and also some which are related to drug metabolism. So again, this makes sense. And it also suggests that metabolism is improving after infection. And again, 2 weeks later, there was a wide increase in a range of different pathways. You don't need to be able to read that. It's just basically a lot of them were linked to carbohydrate and protein metabolism. So this suggests that removing the infection and the subsequent alterations to the bacteria which resulted were improving the metabolic potential of the gut bacteria, and therefore potentially having a knock-on positive effect on the animal. Which is why, I guess essentially this is why we treat animals, because we know that it's having a negative effect, but it's nice to see that that this is directly related to the gut bacteria in this instance. And what's really nice as well is that there's been some studies in mice in experimental infections where they've shown almost identical changes. So it seems that this isn't just specific to the horse, and it's not just specific to the particular parasites that infect horses. So they also showed a reduction in Bacteroidetes with infection and an increase in Clostridia. And they went further on to actually look at the effects of this, and they showed that as a direct effect of a reduction in Bacteroidetes, there was a reduced ability to metabolize carbohydrate and protein in the mice. And they also showed that the Clostridia had an immune suppressive effect. So that was presumably in order to facilitate the parasite becoming infected. So it seems that these changes do have a very real effect on the animal, and it's not just random fluctuations which we can't really relate to. So in summary, impact of infection on worm infections in yearlings on the gut bacteria is to cause a dysbiosis. It potentially has negative nutritional effects through a reduction in Bacteroidetes, and also may have some immune suppressive effects by increasing Clostridia. And so the next question, in my mind, although there's still a lot more work to be done, is there any potential for intervening in this system in order to try and improve our control of parasites and the negative effect it has on the animal? And I think the answer to that is yes. So for the last couple of slides, I just want to talk briefly about how that might be possible. And so there are sort of several different ways that we can alter the gut bacteria, some of which are very specific and some of which are quite blunt. So for example, using antibiotics knocks out lots of different gut bacteria, and we obviously wouldn't be recommending that as a way to control parasite infections, because we'd also be selecting for antimicrobial resistance. There are now some really nice techniques which are being developed in humans and in mouse models, where they can use like almost there's some things called phages, which are like viruses, and they can actually go in and like kill specific types of bacteria. So if you know that you've got a bacteria in someone's gut which is making them ill, then you can send these in and they go and edit them out. So this is obviously quite a long way away from this in horses, but it's food for thought for the future. But what I'm more interested in at the moment is the fact that there are things which we currently use in equine feeds, such as prebiotics, which are substances which we know particular bacteria like to feed on, and therefore it causes a proliferation of certain types of bacteria. And then the probiotics, which are obviously bacteria themselves that we're giving to the animals because we hope that it improves their health and their metabolism. So we're already using these products, but we're using them quite blindly at the moment, and I don't think that I've seen any really firm evidence that they have a definite effect. However, now that we're beginning to understand a bit more about the effects of specific diseases such as parasites on the gut bacteria, it becomes more plausible to be able to actually target feeds and target feed supplements in order to actually have an effect on specific groups of gut bacteria, which we know will then improve the health of the animal. So, for example, we could be giving a probiotic which is based on Bacteroidetes species of bacteria, which is currently are not available or produced by anybody because it's not something that people have thought about. And additionally, it's been shown that some bioactive plant compounds, which are now being fed to farm animals, have some anti-parasitic effect through their impact on gut bacteria, such as flavonoids and polyphenols. But these are present naturally, but we could be adding them into the feeds in order to try and improve metabolism and the gut microbiota in the face of parasite infection. And then this may hopefully improve our overall control of parasites in future. And so finally, just for the last slide, I want to talk about what I'm hoping to do next. So I think firstly, it's important to repeat this work because this is just one stud farm, one group of animals, and what is known is that the gut bacteria are really variable depending on your environment and your upbringing, your development. So it may be that the findings that I found in this study wouldn't be the same on another stud farm, and so that needs to be checked because if we're going to be recommending interventions using feed supplements, then we need to know whether they have to be tailored to specific farms or whether or not they can be used widely. Secondly, I think it's important to actually verify that there is a negative effect on metabolism caused by the changes to gut bacteria, which I've shown in this study, and likewise to do this in relation to immunity as well. And then finally, obviously in the long term, I'll be hoping to design and test dietary supplements based on the findings of this study, but I think that's probably a little way now. But mostly, I think now we'll be focusing on repeating work and verifying data. So if anybody is interested in becoming involved, then we have a big lab at University of Cambridge where we're doing a lot of these studies. So we'll be very interested to hear from people who would like to know a bit more about what parasites are doing to the gut microbiota of their horses. And finally, I'd just like to acknowledge my funders, again, the HBLB, EBF and the TBA, and also my support network and my sponsor at Cambridge University. So my sponsor is Dr Cinzia Cantacesi, who works in humans mainly, but she's been vital to the success of this project. And thank you all for listening. Thank you.